Welcome to today's lesson on mutations. When a cell is copying its DNA during DNA replication, it makes mistakes, and these mistakes are called mutations. Mutations can occur in a greater frequency when a cell is exposed to damaging environmental agents. Agents like sunlight, UV rays in the sunlight, cigarette smoke, X-rays, gamma rays, all of these things damage DNA. Now, cells will catch and repair and fix most of those mistakes, but severe mutations can get through and cause great changes in an organism. Today we're going to look at two main categories of mutations: small-scale mutations like point mutations, and then large-scale mutations that involve irregular chromosome number or huge amounts of chromosomes missing or added in. Now the first is point mutations, and point mutations are changes in one or just a few nucleotides at a time. Substitutions are one type of point mutation, and they're typically less severe. In a substitution, one base is swapped out for another. So here is an example of substitution. You have your DNA sequence, and when we transcribe that DNA sequence, this is the mRNA sequence that we get, and this is the correct amino acid sequence. This is the correct protein. In a substitution, one of those nucleotides becomes swapped out. So instead of having GCA, which you're supposed to have, we have GTA. And then instead of having CGU, the mRNA becomes CAU. Now the problem is that CGU codes for this amino acid, but CAU codes for a different one. It codes for histidine. So in this protein, one of your amino acids is now going to be incorrect. This is called a substitution mutation. The second type of point mutation refers to frameshift mutations, which include insertions or deletions. Basically, you're going to add in an extra nucleotide or you're going to take out a nucleotide. And the reason why this is problematic is because these mutations shift the reading frame. If you remember, the mRNA is read 3 letters at a time. So if you add in an extra letter or take one out, every single codon is going to be incorrect. This can alter the protein so much that it's unable to perform its function. So let's look at a frameshift mutation. So this was the substitution, which we already looked at, and this is an insertion. So here we're adding in an extra letter T, which of course in the mRNA is going to transcribe as an extra A. So we have an extra A in here. Instead of having AUG, we have an AU with that extra A. So our first amino acid is going to code differently. We're going to translate it differently. And the problem is that every single letter here has gotten scooched over. We have shifted the reading frame. And as we try to translate each of these 3 amino acids, we get an entirely different sequence down here. So if you compare these amino acids to these, they're completely different. Here's an example showing you in words, because if we said that codons are kind of like words, so we have some 3-letter words here, that makes sense. The fat cat ate the rat, which makes perfect sense. And here we're doing a deletion, which is another frameshift mutation. We're taking out the letter H, and we're still reading the sequence as 3 letters at a time. So instead of the, we have tef, ATC, ada, tet, her, and that makes absolutely no sense. So this is the severity of a frameshift mutation. Frequency of mutations. Chromosomes in a diploid human cell have about 6.5 billion nucleotides, and we make about 175 mistakes during DNA replication. And that sounds pretty bad, but it's a very small percentage. And on top of that, most of the cell's DNA doesn't actually code for protein. There's a lot of DNA that's just there as filler. We don't know what it does, but it doesn't actively code for protein. So if you make a mutation, a mistake in an area of DNA that isn't being used to make protein, then it might not actually matter. It might not actually cause an effect in the organism. When a mutation occurs in a protein coding region, there's a redundancy in the genetic code also that offers a margin of safety. So what does that mean? Well, this is what it means. Let's say that the normal mRNA is CUC, so that's the normal codon, and this is the mutated codon, CUG. So there's that mistake. Instead of a C, we have a G. But when we look it up, CUC codes for leucine. Now the mutated sequence is CUG, CUG. That also codes for leucine. So there's a redundancy in the genetic code where you make a mistake and it might not actually matter. All right, so for organisms, the amount of mutations that occur are actually very rare and very infrequent. Now, larger scale mutations involve chromosomal mutations. A monosomy is when an organism has only one copy of a chromosome, but they should have two. An example of that is Turner syndrome. A person with Turner syndrome has only one copy of the X chromosome, and I'll show you a picture of the karyotype of a person with Turner syndrome. And a trisomy is when an organism has a third copy of a chromosome when they should only have two. The example is Down syndrome. So a person with Down syndrome has 3 chromosome 21s when they should really only have 2. And I'll show you pictures in a minute. But how do these mutations occur? They occur through something called non-disjunction. So disorders of chromosome number are caused by non-disjunction, which occurs when pairs of homologous chromosomes fail to separate during meiosis 1, or sister chromatids fail to separate. So here is meiosis 1, and at this point, if you remember, this is metaphase 1. These 2 chromosomes should go in separate cells. They should split, right? The light-colored one should go here, and the dark-colored one should go to the right. But that didn't happen. This non-disjunction happened where they didn't separate, and both ended up in one cell, whereas the other cell ended up missing that entire chromosome. All right, so this can cause mutations because these cells, if let's say these are eggs, they're going to be missing that chromosome entirely. And then here, these cells are going to have an extra copy, so that when this egg meets a sperm, that sperm is going to have a trisomy. It's going to have 3 of this specific chromosome. Non-disjunction can also occur in meiosis 2. So far, so good. Meiosis 1 happened properly. This is what's supposed to happen. And then this is normal 2, where you have the sister chromatids separate during metaphase 2. But in this case, they didn't separate. The sister chromatids were not pulled apart. They both ended up in one cell. And this cell, when it now has an extra chromosome, it should have only one, so that when it meets the sperm, it will have 2. When this one meets a sperm, it will have 3 copies of that chromosome. And this one doesn't have any, so when this egg meets a sperm, that zygote is only going to have 1 copy when it should have 2, resulting in a monosomy. So that is how these chromosome number mutations occur. So here's one example, Down syndrome. A person with Down syndrome has a normal karyotype, except for when you get to chromosome 21, they have an extra copy of chromosome 21. Now, Down syndrome is pretty severe. It's associated with an intellectual disability, characteristic facial appearance, weak muscle tone, shortened lifespan, and other birth defects, right? Just because of that extra copy of chromosome 21. Turner syndrome involves having 1 fewer X chromosome in a female. So this is the karyotype of a female because there's no Y chromosome. But females typically have 2 X chromosomes, and this is a monosomy because this female has only 1 X chromosome. A female with Turner syndrome has short stature, they have a delayed puberty, infertility, heart defects, and certain learning disabilities. So this is pretty severe. Typically, monosomies can only occur in the sex chromosomes because these chromosomes, if you have 1 fewer chromosome out of any of the autosomes, that's going to be a fatal or lethal mutation. Okay, so the last piece is chromosomal rearrangements. This is another large-scale mutation. This affects big chunks of chromosomes, but not entire chromosomes. In a deletion, part of a chromosome is lost. In a duplication, there's extra copies of a chromosome. And extra is not good. Just like we saw in trisomies, having an extra chromosome is not something that's a good thing. And then inversion, reverse direction of parts of a chromosome, and in a translocation, 1 chromosome breaks off and it attaches to another chromosome. So that's a translocation. And here are some pictures of what those chromosome rearrangements look like. There's the original chromosome. Here's a deletion where we're missing a part. Here we've duplicated a part, inverted part of a chromosome, and translocation where a chromosome has detached and attached to another chromosome. Here is an example of a chromosome rearrangement mutation. It's called Cri-du-Chat syndrome. And what happened here is here's the individual's chromosome number 5. This is normal. This is this pair is normal. And here, part of chromosome 5 has become deleted. It's missing part of chromosome 5. Infants with this condition often have a high-pitched cry that sounds like a cat. This disease also comes with intellectual disabilities, smaller head size, and widely spaced eyes. So just missing a chunk of a chromosome has a very severe effect on the individual. So I hope this was a helpful introduction to mutations. Thank you so much for listening.